Good afternoon to all of you. Let's get started because I know time is always pressing on busy faculty. My name is Walter Edwards. I'm the director of the Humanities Center, and I'm delighted to welcome you to this talk by Professor Noah Ofen, who is an associate professor in the Department of Psychology. And she joined that department in 2011 and has hit the ground running because after she joined the department, I think she joined the department as a faculty associated with the Institute of Gerontology and the Merrill Palmer Skillman Institute, but also she was associated with the Department of Pediatrics, and then also with the Department of Psychology. And before coming to Wayne State University, I'm just deeply impressed about the quality of people that the Wayne State is able to attract. And she was a postdoctoral fellow in psychology at Stanford University. She was a teaching fellow in the department of neurobiology at Harvard University. At Northeastern University in Boston, she was a lecturer in the Department of Psychology and the Massachusetts Institute of Technology. She was a postdoctoral associate in the Department of Brain and Cognitive Science, and so on. She, of course, got her PhD from the famous Weizmann Institute of Science in Israel. She got her PhD there, and also she got her master's in science from that institution. She graduated magna cum laude from the University of Haifa in Israel as well. She investigates structural and functional brain development across a wide range of ages, typically developing children and adults, using tests of cognitive abilities combined with neuroimaging techniques. Dr. Ofen probes how brain structure and function shape human cognitive functioning across development. She focuses on individual differences as expressed through behavior and biological measures. These include brain measures and genetic polymorphism. She works to explore the structure and function of the hippocampus, a crucial brain structure for learning and memory, and uses innovative methods to investigate neural activity that supports memory in pediatric epilepsy patients. She is absolutely well published. And I wouldn't read her list of publications because that would take up too much time. But just last year, she published 2 pieces along with a group of her colleagues. One was differential functional connectivity in anterior and post-anterior hippocampus, supporting the development of memory formation, published in Frontiers in Human Neuroscience. And also this year, that year, she published with a group of scholars, cumulative antenatal risk and kindergarten readiness in preterm born preschoolers. This was published in the Journal of Abnormal Child Psychology. So we're in the presence of a very busy and very, very well-known international scholar, and the Humanities Center is delighted to welcome her to give a talk today in part of our Brungback series entitled Memory and the Developing Brain, from Description to Explanation. Thank you, Dr. Ofen.
Wonderful. So I guess the slides were up the whole time. Let me just stop share this so you can see me as well, and say thank you for a very kind introduction. And it's really a pleasure to see all of you. I know that it's a wide range in the audience, and I'm hoping that all of you would find some aspect of it related and interesting, stimulating. I'll be happy to answer questions at the very end. And again, thank you for the very kind invitation, kind introduction, and for inviting me to be part of this series. So let me go back to sharing the screen and make sure that you can all see the screen now. Am I correct?
Yes.
Yes.
Perfect. Wonderful. So today I'm going to talk to you about memory in developing brain and really show a little bit of the work we've been doing in my lab over the past few years, or in general, kind of the scope of the research program that I've been conducting, and trying to kind of make the jump towards the next big step. And the next big step is continuing to doing the work that we do with the more focus and trying to understand how can we get all the measurements, all the types of available new methods that we have, tell us something new that takes us from just going and describing the phenomena that we see into maybe something that's more explanatory, that gives us a kind of deeper understanding. And okay, great. I always like to start by showing pictures of my kids, and it changes every time because, you know, they grow. But what I like to say is that a child comes to the world with very little ability to interact with it. And over the unfolding of many developmental milestones and developmental processes, they learn to walk and talk, run and climb. Through more formal education, they learn to read and write, do math, maybe play chess and play some music, and maybe get to the apex of existence. This is engage in cognitive neuroscience. And underlying a lot of those developmental processes is memory. Memory is integral to all of those aspects. And it was, to me, quite interesting when I started my postdoctoral training that at this point, given how much we understand memory as being crucial to development, there was very little research to understand the neural basis of memory development across the lifestyle. In my lab, that's what we do. We kind of tackle memory development with a cognitive neuroscience approach. And it's really kind of keeping an eye and making progress. And this is a little bit the structure of the talk, but looking at the behavior, trying to quantify memory, to understand better what is memory. Understanding of what is memory is crucial for us being able to do anything in terms of describing it, in terms of bringing those other measures from the brain. And in those measures in the brain, we kind of, a gross distinction would be between looking at how the brain functions with the methods that we have to track brain functional activation or functional activity. This would be functional MRI and intracranial EEG. These are the methods that we use that I'll present a bit of our work with later on. And looking at brain structure. The brain changes over development. And when we look at brain structures, we're trying to quantify those structural features, say, for example, the volume of different regions in the brain. And we're trying to understand how those structural features that we can quantify are related to the development of cognition, in our case, memory. In a way, we're thinking of brain structure, as I highlighted, is this endogenous constraint. This is what limits and enables the increase in the range of cognitive abilities. And back to the brain functions, I'm thinking of those more as capturing the neural correlates. So you learn something, you remember something, how did the brain, what was the brain doing? What function, how the brain function in order to, what are the correlates of those abilities? So this is a brief introduction to my overall thinking. And as I said, a little bit of how I structured this talk. And as you can see in these plots, I'm not the only one interested in those, or our lab is not the only people. As you can see, along an increase in behavioral investigations of memory development, the use of neuroimaging techniques to probe memory and its development has risen sharply in recent years. This increase in the prevalence of studies of brain and behavior conducted with patients or participants across a wide age range holds the potential to uncover mechanisms of developmental change, prompting a timely evaluation of outstanding inquiries and best practices in the developmental cognitive neuroscience of memory. So we're trying to see how the fact that we have so much more that is produced, that is out there in the field, what are the opportunities that we now have, again, to go from just getting a snapshot description of what we have and into really deepen our understanding of the mechanisms that govern both the correlates and the way by which the brain changes govern cognitive change. As I said, we'll have three parts, kind of roughly along those behavior, function, and structure. And the first part is kind of something I put a little bit more effort here for this specific forum. And it's really giving a broad thinking of what memory is. And I've titled it, What is Memory Development, the Development of? I'll say a few more in the next slide. And then I'm going to go into showing you a bit of what we have learned by looking at the neural correlates of memory, the new opportunities we have, and a little bit kind of critical brief note about the fact that we still have limitations in our measurements, and we always need to keep this in mind as we continue to strive towards being able to interpret the findings that we do have. And then the third bit, we're going to do a similar approach with looking at the brain structures. Again, what have we learned? What are the opportunities that we now have, and kind of keeping a note of the limitations that we have as we strive to get, or what is it that we still lack, and how we can define new questions to get at that based on the available methods that we have. So I said we're going to start with what is memory development, the development of. I always say that it's just a good opportunity for highlighting that it's not because English is not my first native language that I say such funny sentences. This is actually the title of a very influential Society for Research in Child Development symposium to the date 50 years ago. So in 1971, that was the title of a very influential at the time a symposium. And if you looked at the slide that I've shown you about the number of studies, you actually would have seen that in terms of memory behavior or memory development was a little bump right around this 71 to 75. This is an era where there was a lot of interest. And the idea in this symposium was that we want to understand memory development, but memory development might likely reflect a whole. It's an interacting, as a child is developing, many things are changing. And when we try to understand memory development, we need to kind of try to start defining the scope and defining the boundaries of the phenomena that we're interested in exploring. And specific three big other things or related things that have been highlighted in what is memory development are the fact that there's increasing the ability to keep more information in mind. This is a capacity. There's a growing knowledge base. So if you remember more things, you might be able to remember the relationship between things better because you know them better, right? There's knowledge base. And the last would be strategy. That you're more aware of different types of mnemonic strategies, and you're better able in utilizing those strategies. So all these are interesting, and although 50 years ago, are highly influencing the way we are studying memory development. In my talk, and for us, we really kind of try to just know that all these are happening, and some of our studies are geared to kind of look at the influences of those which we might think kind of support elements for memory development, really try to understand this first part, what is memory development? And again, development of, we would actually bring in our cognitive neuroscience methods to say it's the development of the structural features of the brain, and it's development of the ability of the brain, the way the brain functions, right? So this is kind of bringing a new method in order to address this, what is memory development. But before we get to memory development, we really kind of need to spend a couple of minutes maybe thinking what is memory. And this is really important because if we don't have very good or very specific theoretical framework to understand, to kind of what is it that we're talking about memory, then it's going to be very difficult to get to ask what is memory development, how would the child, what is memory in the child, and how that might differ by age. And this is also particularly important when we try to get this cognitive neuroscience approach. And in this case, when we're bringing the brain, and the cognitive neuroscience approach is really what cognitive neuroscience do is really trying to understand how the brain gives rise to the mind. The brain is composed of many different things that we can think about them in different levels of organizations, right? So we have the whole brain on the central nervous system, but those are all really can go down in the level of organization, and anything that we do that we think the brain does exists across all these levels of organization. We focus on the top levels and we're trying to link some things to behavior, and oftentimes this can be done by doing this level of analysis approach in which we try to understand what are the computation, kind of what is memory, how it might be implemented in an algorithmic way, and then how it be implemented specifically along those levels of organization. So all of these are important, but we can't get anywhere forward. And I brought this specifically for the Humanities Center because of like the philosophical flavor of those. What is memory? And we need to know what is memory in order to start to understand how it developed, and in order to try to link it into the brain. So what is memory? I'll just this slide I'm just going to show with those of you show with you the kind of textbook, still pretty much a textbook description of memory. We know that it's not a unitary thing. There are many different type of phenomena that would be memory. And broadly, it's thought that there are memory that is non-declarative, just how to ride a bike. And the declarative memory, they think that we know that are memory for facts and events, and thought to be accessible to consciousness. And within declarative memory, there's been a distinction between these facts, semantic memory, and events, and memory for specific things that happen at a certain time in a certain place. Although development is crucial for all of those, in my lab, we primarily focusing on episodic memory. But when we think about memory, what I showed you now, this is more of a static or kind of storage-based account. But of course, memory is very dynamic. When there are different stages in the life of a memory, information is being encoded, somehow in the brain it's being consolidated, and then upon request being retrieved. In this dynamic perspective of memory, we can think about the different processes or operations that are happening in each and every time, every stage of the memory. And those operations could take different forms. It could be rapid operations that generate flexible associations that we can keep in mind. And that might be what happened in episodic memory. Maybe more slow and rigid associations is what we need to generate knowledge about facts or about specific procedural skills. And there yet be maybe another rapid yet inflexible information that happened, for example, when we see something, the next time we see it, we actually already respond to it quickly, and it doesn't need to be something that is very flexible, right? So those are different types of memory phenomena or memory systems. One might say that, as you can see, is parsed out different than the way it is. And those current discussions in the field are really exciting and call for us to reevaluate the boundaries of this, the boundaries of the phenomena that we are investigating. So I'm not going to say too much about this, just that right now, all those are influencing the way we think, the way we decide which questions we're going to be following, the way we understand how to interpret developmental dissociations that we are finding. Is it between declarative versus non-declarative, or is it perhaps between the types of operations versus the nature of representations in the memory that we are following? So with this, I can start, all this is in the background. Now we can go into asking what is memory development. And a synthesis of a lot of mine and other work in the field has really suggested that if you can think about within this episodic memory for past events or experiences, there might be the best way to capture developmental dissociation is if we think about the richness of the content of our, or the richness of representations that we are referring to. And if you think about a rich representation, it would be rich in specific details, might be associations between items, might be associations of item-specific context, might have the phenomenology of I really remember. I can do this mental time travel, again, the hallmark of what would be episodic memory. Those are the, and this could be contrasted to some type of memory that is more low in details. Maybe it's a single item rather than associations between items. And maybe it has just a sense of, it just feels familiar, rather I can really go back and recall a very rich memory of this past experience. And I'm kind of spending time there because this is what I've promised you as developmental dissociations, at least within the age range that I've been focusing in we've been focusing in our research is between middle childhood and young adulthood. It's those rich association, detailed memories with associations and this phenomenology of I really recall or recollection, past event that show a marked increase with age, whereas there's much earlier maturation and first stability within this age range for types of memory that don't require this high level representation. And this is just showing how do we get to do it? We get to do it by, for example, this was schematics. Now let me show a little bit of data here. If we ask people to try to remember pairing between two word pairs, and we ask them to then either remember just a specific word or the exact pairing where these two go together, then we find indeed that it's the memory for association that shows this increase with age, but just remembering a single word seemed to be fairly stable in the kid as young as 5 compared to a kid as young at 8, compared to our young adults. So this was one example with one task. A lot of the work we do in the lab now trying to get this a bit more generalized. And for example, this is work from the dissertation of Shijing Liu in the lab that has taken a lot of tasks, put them all together, try to get those latent constructs of this item and the associative memory, and then look for the age effects there. And when we take this approach, again, we see this is for remembering associations or more a high dimensionality in the representations versus just remembering something of lower dimensionality, just remembering in this, for example, we can term that item memory. You see that there's might be earlier on still increase, but it flattens and plateaus. So some aspects still show protracted development, some seem to be maturing earlier. And this is true not just for representation, this is the utilization of strategies. So this is the older you are, the better you are utilizing semantic strategies when you recall information. So this is just a sample to show you there are some aspects that increase, some aspects that are fairly flat. And when we go to look for the neural correlates, it's important to know we would try to understand what is it that accounts in the brain for those increases and that go with it. So what is memory development, the development of? Once we have a slightly better idea, we can go back and get into defining, trying to identify where is it that we have changed and then go and look at the neural correlates of memory development. how, when we get to these neural correlates, there's really two big questions that we try to answer. First was where. Again, this is my brain. So right here, there's regions in the brain that we know are relevant for memory. Is the way that they are active, or the activation of those regions, differ by age? That would give us a sense about, again, there's neural correlates of memory development. And the other, much more interesting and kind of the holy grail for our investigation is really how. How does the brain region support age in a way that show this age increase in memory? Once we know how, again, going more towards this mechanistic understanding, we'll be able to do a lot more because we have a way that we can infer some causality, for example, rather than just showing what it is that the brain is doing. So going into this, where first our investigations are focusing on the prefrontal cortex and the medial temporal lobe, including the hippocampus, two regions that are critical, important to memory. It's crucial. The involvement of the medial temporal lobe was specific, the hippocampus. Global lesions in the hippocampus result in global amnesia. This is the inability to remember any new facts or events. So it's specifically, global amnesia is this nature of, specifically for this declarative memory or episodic memory. But in addition, there's also important involvement of the prefrontal cortex, where the effects of memory are more subtle, but they specifically relate to those more rich representations that I showed you before. Knowing the specific associations, knowing the who said something, not just what was said, those are aspects that are critically supported by the involvement of the prefrontal cortex. So this is where we're going to look for, and how we're going to get, how do we get into trying to get activations that differ by age? We use functional neuroimaging that allows us to get a snapshot of what the brain is doing when it learns information and subsequently can remember that. Or when it retrieves that information upon a request call, and perhaps even how this information is being consolidated or stored. In our work in the lab, we focused more on this encoding and what is it that the brain is doing when information is being learned that predicts that this information is going to be retained. Okay, so here is an example, and again, we're going to use this functional neuroimaging to see what the brain is doing and which of what it does differ by age. So we use this to look at the encoding. We use a subsequent memory paradigm. It's simply to say we get participants into the scanner while we monitor the brain. And we preferred stimuli in the lab is scenes, pictures of scenes, and there will be indoor and outdoor scenes. We simply ask participants to look at each of those scenes, tell us if it's indoor and outdoor, so we know they actually looked at it sufficiently well, but also try to remember them. Again, all this while we monitor the brain, we take them out of the scanner, and then we give them a recognition test with all the pictures that they've seen before and new pictures that haven't seen. Some of the picture that they have seen before, they would correctly say, I have seen it before and I'm sure I've seen it before. So we ask them something about their phenomenology or the strength that they feel regarding how sure they are and how confident they are that they've seen before. So some they'll say, yes, I've seen it and I'm sure. Others, you can see this one, they will actually sort of forget because as far as we're concerned, they forgot because they say, nope, I have not seen it before. This is a new picture. What that allows us to do is go back into the brain activation that we have, the brain images that we have, and sort it such that we can distinguish the time points where there was a scene that was later remembered versus a scene that was later forgotten. Okay, so I'll show you what the brain does in a second. Just before, I'm still going to show you for this specific task, this is an example of how they behave. Marked increase with age. Specifically for those that they said, I do remember seeing it, I have fairly high confidence. So we're going to look for that specific increase in behavior. What aspects of brain function support that increase in behavior? And this is for the first time I've done it over a decade ago. in which we, again, this is what the brain was doing more so when an item was later remembered compared to later forgotten. And you can see here regions in the prefrontal cortex and regions in the medial temporal lobe. Okay, this is of course everyone. What differed by age, you may now ask, thinking to yourself, because I kind of led you there. Not so much within the medial temporal lobe. Remember this, I said the region that's most crucial to memory does not show very robust differences in the overall activation with age. It was a prefrontal cortex activation that seemed to be more tightly related to the increase in this ability to form memory. As you can see from the children, adolescents, and adults, there's increase in within these subgroups in the way that the prefrontal cortex activations was related to subsequent memory of rich memory. So we can already conclude way back that it's likely the protracted maturation of the prefrontal cortex that's related to age increase in the ability to form and retrieve, this is from other studies we've done, rich episodic memories. So this focus on the prefrontal, this finding really kind of made us want to know more about the prefrontal cortex. And in more research studies in the lab, led by Ling-Fei Tang, now a postdoctoral fellow in the lab, we continue to collect a lot more data with the same task that I've shown you before, but now we are focusing on, in this study, our investigation on the prefrontal cortex. And with a much larger group of participants, we still identified those positive regions. This is more active when you later remember than forgotten, but we also find a really interesting, this blue blob over here. These are regions in which activation was showed the opposite effect. And it was a very specific opposite effect. So for the positive, if you see this more active for subsequently remember than subsequently forgot, for the blue regions, we got an increase in the level of deactivation. They were more deactivated when the item was when the scene was subsequently remembered compared to forgotten. So now we have like complementary aspects of the prefrontal cortex. It's not just the positive, there's also some negative, but it's really the dynamic way by which the brain functions that supports subsequent memory. And these are important, both of them, we found out, to development. So if this is what I've shown you before, the older you are, the more you do this subsequent memory increase for the hits compared to the misses. It's also for those negative, we find with ages increase in the deactivation compared to in the deactivation for those that are subsequently remember compared to forgot. And even more interesting for us was that part, this negative, seemed to be more directly related to performance, because it was the variability in this negative subsequent memory effect that partially accounted for the age-related increase in memory performance. What are those regions? They might be related to this ability to do something like a thought suppression. So we have a lot more thinking about it, a lot more ideas what it might mean. And of course, at this point, these are just our thoughts. So now that we have kind of prefrontal, and even more so, we know a bit more about the different types in which the prefrontal cortex is supporting, we can start thinking more about the how. And one way in which we can start addressing how is by looking not just as where it is, it goes up, it goes down, but the interconnectivity between regions in the brain. And for this, we use an approach within fMRI that allows us to look at the co-activation, the covariance patterns between regions in the brain. And we use this approach to look at seeds from these two different regions in the prefrontal cortex. And we find that with age, those regions were covary more, were more similarly active with the medial temporal lobe. Both the positive and the negative with age were more connected. So now we can think it's more about the medial temporal lobe connecting with the prefrontal cortex, although the medial temporal lobe didn't show robust differences. It did once we looked at these connectivity patterns. in the most recent study, we took a different approach and said, okay, within the medial temporal lobe, we can look at anterior versus posterior, and then look at the age differences that support subsequent memory over age. And we were able to pinpoint more specifically that it might be the refinement with age of patterns of connectivity in the posterior hippocampus with both those regions in the prefrontal cortex, and not so much the anterior hippocampus. All those are placed, and I don't have time to get into it, in larger theoretical perspectives of anterior versus posterior memory systems. But right now, we're just kind of getting a little bit into the how. How might be refinement of regionally specific medial temporal prefrontal cortex functional connectivity. So kind of interim summary of this part 2 for this, we have something about what we're looking for those regions, where maybe more so the prefrontal cortex than other regions in the brain, and how by its connectivity patterns with the medial temporal lobe. But I kind of said, okay, that's what we learned, but what we should be looking for, and it's really the fact that we kind of, this functional MRI measure don't give us a very precise measure. Enter our better ability to look into how the brain regions support age increase. We really want to get the spatial temporal dynamics of brain function, and we can do that with a series of studies that we have been analysis that we've been working on by taking advantage of data recorded directly from the brain. These are epilepsy patients. And part of their clinical management, they have implanted electrodes as part of brain mapping, pre-surgical brain mapping. And what that gives us is direct way to get into the activity in the brain that is localized. It gives us all the richness of the temporal effects and all the richness of the different frequency ranges by which we can probe that have been that have been linked to different potential brain mechanisms. And the first, and this is just to kind of, we are very excited about this work, as you can see, looking in our screens. So I'm going to give you two brief introduction to two of the studies led by Lisa Johnson, the first one, who is leading this work in the lab now as was a postdoctoral fellow at Wayne State and Berkeley, and on the job market and doing phenomenally. So congratulations, good luck for this. So first study that Lisa led is direct brain recordings that from the prefrontal cortex. What you can see here is an aggregate of all the electrodes that we had across, in this case, 17 participants, 17 patients. And now we can start to look, remember I showed you a lot of the prefrontal cortex, we can start to look at what are those different regions do, how does the activity in them look like? And again, I'm gonna go very quickly. Just to say, we focused here on the high frequency broadband. This is just a wide range of frequencies that have been linked more so to the prefrontal, to the fMRI activation. So we're trying to get a bit more of the timing, but something that might be related to what we already know from functional MRI. And this is, as you can see here, from one of the regions in the frontal cortex, an increase in power for hits and a similar increase in power for all those trials that were misses. But we now can see the specific timing of this information. Overall, as you can see, the hits and the miss are pretty similar. The subsequent memory direct contrast of hit versus miss across the whole group wasn't very telling. There wasn't very robust effect. But what we did find is in each of the participants, there were electrodes that showed us significant subsequent memory effects. Those subsequent memory effects of positive, as you can see here in the yellow, or of negative, as you can see here in the blue. Those effects were across different electrodes in different frequency ranges and in different temporal positions. So the bottom line that we have been able to see, even in a very young participant, and as in our old participants, there was a lot of diversity. This is was true for this one region that I showed, but this is really the case across all different regions that we identified in the prefrontal cortex. So just from that bit, we conclude the spectral subsequent memory effects are diverse within the individuals, but they're similar across children and adolescents. So the fact that young children show memory relevant frontal activity patterns that are similar or like those that are observed in adolescence to some extent challenge this prevailing theory that I've just kind of put forward. It's really the prefrontal cortex that protracted development. Of course, there's a lot more that we have done and will be doing. For example, now we can say that this diversity is similar, but there are much more subtle effects, and those effects and the timing of interaction or activity flow between regions in the brain that dictate what is it that we subsequently remember. A lot more to do, and we're specifically focusing on looking at the crosstalk between the medial temporal lobe and prefrontal cortex, as well as additional aspects. So this was 1. 2 is a study led by Xin Yin in the lab, and she was really interested in talk to you up until now, the medial temporal lobe and the prefrontal cortex, but really when we see visual information, there's an important role for visual cortex. So she was really interested in looking at effects within the occipital cortex, and we had electrodes there, and this is a description, the distribution of their locations, and from the scenes that we have, Xin took advantage of the fact that we can look at something that's more related to their perceptual description. And this is by looking at scenes that are of high complexity. In this case, they have more than 4 unique object categories, or low complexity, 3 or less unique object categories. Now that we look at those distinction by high complexity and low complexity, we're doing it not just to get something that's similar to scenes, but also because in our past study and confirmed here, we found out that the age difference in memory, remember I told you sometimes it's stable, sometimes it shows more robust effects. It seems that the age difference in memory is specifically pronounced when we look at the high complexity scenes. The older you are, the better you are in, but specifically those high complexity scenes, not the low complexity scenes. How does the brain support this? With a focus on the occipital cortex and on the alpha frequency, which is a powerful, robust frequency that has been investigated in effects looking at the occipital cortex. And specifically, what happened within this alpha frequency range, if you look at the alpha power, there is a decrease in power following the presentation of a stimuli, thought to reflect this kind of also termed or referred to as desynchronization, so thought to convey or carry sort of the information, the visual information. What you can see here for the hits compared to the miss, there's maybe a little bit more of those those alpha power desynchronization. And indeed, this is the case when we contrast those two across all participants. But as you can see, it's much more for the high complexity scenes compared to in the low complexity scene. Following up on the age differences, looking for age by complexity interaction in this alpha frequency were significant. As you can see here, this decrease, as I referred to earlier, desynchronization, was this desynchronization increased with age, and specifically for these high complexity scenes. More so, it was predictive of overall individual differences in memory accuracy. And again, among those of the older participants that we have here, the more desynchronization, the better they performed. So now we have information from within the occipital cortex that directly relate to how is it directly relate to memory. But what's and this alpha power effects differ by complexity and age, and correlated with age-related increase in memory for high but not low complexity scenes. So like getting us a bit more about, again, we have when, we have what frequency range, we have a lot more rich information, but it doesn't end there, because once we look at the alpha frequency, already just that within this one frequency range, we can tap at simultaneously at something completely different that might be happening in terms of the mechanisms by which information is being carried in the brain. And this is what Xin was looking at, the instantaneous alpha. This is the actual frequency. And it turns out that the older participants were, specifically for the high complexity scenes, they ramped up the alpha frequency. So they kind of had more cycles within the same time domain. That aspect that we found that differed by complexity and age was not related to memory. so perhaps we think it relates to age differences in perception, but I'm showing this just to give you a sense of the richness that we can get from this type of information once we start getting this rich direct intracranial recordings from individuals. Again, a lot more to do there that we are pursuing, but just to summarize now, we have we're starting to know where, we're starting to know how, and there's a really rich set of questions we can start asking about the mechanisms that support memory in the brain. Just to keep in mind, I said we'll end with limitations, each of these bits. We do need, especially for fMRI, to be mindful, well, for fMRI and also for the behavioral measurements that we are involving, we need to keep in mind that we have reliable measures, and this is work from Ling Fei right now, looking at the same individuals coming back twice. And now we can look at the brain activation in time 1, brain activation in time 2, and see how similar those brain activations, after we also assess how similar the same individual's performance on the task that we chose to use. And we can see that there's a relatively high, still a lot more to crave in terms of the intra-class coefficients are within a good range. For the whole brain, for the group level, also it's in the good range. And within each individual, though, we do have some variability, and especially once we, and there's some hint that perhaps among older participants, we have better reliability than younger. Those are things that we continue pursuing to know better our measurements, and also to be able to understand where can we be more confident in our interpretations versus not. So this is all was part 2, and at the interest of time, I'm going to go fairly quickly through this part 3, which is another line of work in the lab, still all related to, we're trying to understand how brain development relates to cognitive development. But now we're focusing on the regions of the brain and trying to and bring in those more structural gross measures of brain integrity. So looking at the neural substrate of memory, again, the brain structure, same idea. What have we learned? What should we be looking for? And it's similar questions. Which are those brain structure features that differ by age and related to memory? And then how do brain regions support age increase in memory? That how we kind of really just starting to get a little bit to, but there's much more that we need to crave in order to be able to start addressing it more robustly. But where are we looking for? Again, there will be the prefrontal cortex and the medial temporal lobe, or specifically the hippocampus. A lot of work from many labs demonstrated that the brain continued to change. Longitudinal studies have shown structural changes in the brain. In fact, somewhat counterintuitive, not get to it much, the brain, the cortex tend to thin down between middle childhood through young adulthood. And this is what you can see here, it's kind of getting bluer, it's kind of get thinner down. And this is done in different rates and most protracted within the prefrontal cortex. Again, link into all why we thought prefrontal cortex is a key region for showing us the age differences in memory. The other region, the hippocampus, globally doesn't seem to show much differences by its volume, but there might be some regional differences in it that people have, including ourselves, have been trying to look at. So let's start quickly with one example of the way we can link prefrontal cortex variability to memory, and we'll look for. I told you prefrontal cortex is more those subtle effects, perhaps the ability to implement mnemonic strategies. So we'll ask this question of when we link the prefrontal cortex to memory. And for the hippocampus, we just want to ask, because I showed the overall doesn't change, but there might be something in the regional changes within it that relate to memory. Those are all active questions that we are pursuing. So starting with one example from Sijing Yu and Dana McCall, in the lab, they were asking looking for the way we can link prefrontal cortex to memory, and in this case, as I said, the utilization of mnemonic strategies. And I'll go quickly through the details. So I'll kind of just put this all up and go through this. Individuals were giving a word list, and then within this word list, words were chosen from different semantic categories. When they gave us the output, we asked them, recall as many as you can. What we did is we went and saw how many times did they pull out words from the same semantic category to put them one next to the other. Why? It's a good strategy. When you try to remember something, we put things that are semantically related. It might help you pull up the other thing that is semantically related. So this semantic clustering is what you see in the field circles. I've shown you this snippet earlier. Increased with age. The older participants were, the more they use this very useful mnemonic strategy. Now we wanted to find that, does that relate to any of these gross measures in the brain? And indeed, this is how much semantic clustering that was related to specific volume of this specific region within the right prefrontal cortex. More interestingly, we identified that indeed this the volume of this prefrontal cortex was partially, again, accounted for the age-related increase in semantics clustering. So we have now a gross measure in the brain that kind of tells us perhaps something about the limits that the brain impose on our ability, or young children's ability to use those kind of useful mnemonic strategies. So this is one example, but most of the work in the lab, or a lot of the work in the lab, is focused on the hippocampus. And when we look at the hippocampus, we are trying to understand how it links to performance, but we also look at other things. So this is from Shijing Yu, just a snippet of why hippocampus is so important. This is actually not, put aside memory for a second, socioeconomic status that differ across individuals. Within a group of children, we found that the social economic status as composite score of cross measures that we had was positively related to the hippocampus volume. Those with a higher socioeconomic status had larger volumes of the hippocampus within the sample that we had. Others have shown additional more effects to the same extent. So the hippocampus, in general, everything, it's kind of side note we do is interesting and important, and has real potential influences in real life. So in the last couple of minutes, I quickly just go through this this bit of the talk. When we look at the hippocampus, I said overall not much, but it has a complex anatomy and complex function. And specifically, this hippocampal subfields, the different subfields of the hippocampus that has been linked to specific memory aspects. So the question we're asking when it comes to the hippocampus is, are age differences in hippocampal subfield volumes related to age differences in aspects of memory? So in order to answer this question, we say, first, are there age differences? And second, do those variability in those relate to variability in cognition? But before we go there, we need to do our work and come up with good measures. And this is where advances in neuroimaging allows us to really get at that level of hippocampal subfield, which is very exciting. There's work that was started in the lab by then a graduate student, Anna Doherty, who is now, I'm very happy to say, a phenomenal colleague as an assistant professor here at Wayne State University, who's really championed this work, and not just within Wayne State, but really led international efforts to get that measurement of hippocampal subfields harmonized across all labs in the world that are interested in this measurement. So in our lab, we started by kind of using this, and what you can see here, this manual tracing among radars with established high reliability, and I won't get much into it anymore, but just to say now we had initially some measure, we again keep working on it, keep changing this, but now we can go across all of our participants, and even ask colleague, in this case Naftali Raz, who had a lot of data from older adults, and we can come up and ask and use the same method across a large wide age range to see that indeed we find age differences in hippocampal subfield volumes. And just within the individual differences within the kid data, using the task I already showed you that shows that specifically for associative memory increase with age, and together again with Dr. Doherty, we were able to show that the increase in associative memory with age was partially accounted for by the age differences in specific region of the hippocampus, the CA3 dentate gyrus, which makes a lot of sense to us because this is one region that we more specifically related to associative memory. So I'm going to skip over the next slide. And just to say we get reliable measures, I told you, with our age differences in the subfield, and those relate to memory. Many next steps here. For example, more work to harmonize those methods, but particular, and this is kind of what we're still missing, is longitudinal data with which we can look more carefully in those effects, and really just with only with longitudinal data we will be able to link to ask anything about developmental change, because to say change, you have to have some indication that we're talking about the same individual trajectories. And you cannot think about change if you only look at cross-sectional data, yet in our field, cross-sectional data is a primary source of evidence. So more of the harmonization methodological tools. This is where I said Dr. Doherty has been champion and recently a post-doctoral fellow in the lab. Kelsey Canada has kind of jumped in this. Xijing Yu, Roya, they're all kind of continuing those efforts. And I mentioned we need the longitudinal data. This is just a nice way, one snippet to show you. This is 3 participants in our study at time point 1 and their brains at time point 2. And you can actually see how similar those are across the years. And then with our good methods, we can actually start getting into, with longitudinal data, we can get to start get the sense to have sufficient reliability in our measurement, so that we can look at the change within individuals and how those relate to cognition. And to this extent, a recent effort led by Roya Homayouni in the lab have directly established the reliability of those methods that we use. So it's not just this high resolution and the fact that we're manually doing it, but in the specific protocols in which we look at what happened between visits, we could demonstrate that we get really high reliability. With our methods, this is not, our measurements are not biased into whether it was measured the first time, the second time. And we have quite a lot of spread in our estimates of change between time 1 and time 2. This is example of what we see over 1 month, just as a retest reliability. This is over 2 years. And this is a spread that we have over 2 years, giving us hope that we can link those variable change estimates or change measures into the change in cognitive abilities, once that we know how to monitor them well. So with this, I'm just going to end by I took you through this little marathon of like starting with what is memory, what is memory development of, and then saying we're really interested in neural correlates of memory, and we have different types of them, and we kind of taking advantage of opportunities to move forward as best we can. And then looking at the neural substrate of memory in the same, taking advantage of opportunities in the field to make the best. And of course, I can end without thinking. I've tried along the way to kind of show you the faces of some of the people who are more crucial for this work that I showed you, but really it's a joint effort of many, many, many individuals in the lab. Those are a list of undergraduates in the lab. Some of them are pictured here that have really done a lot, made really incredible contributions to specific aspects of our work. And a whole list of collaborators now and over the years that have really helped me with which it's really fun to work and produce kind of more questions, not just about what I'm interested, but a whole range of other related topics. With this, I'll end, and thank you very much for your time.
Thank you so much. I think we have a little bit of time and we can stay after. If anyone has any questions, feel free to raise your hand or unmute your mic. And if you just stop sharing your screen, maybe we can see. Yeah, perfect. We can see if anyone has their hand raised or any. All right, last call for questions. If we don't have any, oh, yeah, you can unmute your mic, Laura.
Hi, Noah. Thanks for that talk. One question you mentioned at the very end, the limitation of the scarcity of longitudinal studies. Are you aware of any longitudinal studies within the topics that you addressed that are currently going on? And how feasible is it to do one? I mean, of course, I understand all the difficulties logistically in doing a longitudinal study, but I was just wondering if you could say any more about that.
Yes, thank you, Lara. This is an excellent question. Yes, there are. Our efforts have always been to incorporate those longitudinal findings in very large studies, such as ABCD. This is a very, really, really large study in which the goal and the successful N was at the range of 11,000 participants, to be followed every 2 year for 10 years. Those studies are exceptionally well suited to address specific questions. But for example, the level of, as you can see behind me, this is a specialized high resolution image to capture, with which we can really capture those hippocampal subfields. That type of measurement is not included in those type of studies. Right? Go back to the other opportunities that I mentioned with this really rare opportunity to record information directly from the brain of individual patients as they undergo brain mapping. This is not the type of study that we can generate this longitudinal sample from or a very large sample with. So there's like two different aspects. We want it longitudinal. We also, there's value in the numbers, maybe even if it's not just longitudinal. So there are a lot of efforts. They will be excellent in producing answers to the questions that they've been designed to address. So this large study that I talked about, the ABCD, it's really about, it's really more of a, I think, I forget exactly how it's defined, but it's trying to understand drug abuse and other environmental factors that might be influencing. Of course, now with COVID, everything changes. So now they can ask the effect of COVID on brain development with the limitation that it's a cohort effect. They always collected the 9 and 10-year-old, now I think 12, 13-year-old. So they had COVID at a certain time point. So yes, there are longitudinal effects. We have some longitudinal data. We have efforts through the work of Kelsey Canada now to bring data from other labs that have collected longitudinal data to do a smart data integration to be able to kind of take advantage of the longitudinal. So it's feasible. It takes some creativity, but there still will be a lot of questions we won't be able to fully address yet. And it's fine. Maybe not all the questions are equally important to address now, but we're hoping that we'd be able to.
Great. Bruce, I think you had a question. Feel free to unmute your mic.
Okay, thank you, Dr. Ofen. That was very interesting. I have one comment that philosophers distinguish between 4 kinds of knowledge, and I think there are analogs about memory. So there's that, which you had the memory that, and it had then 2 subcategories. And knowledge how, you can, that's like memory how, it's kind of skill memory. There's these other 2 that are knowledge of what it's like knowledge of what it's like to feel sad or to see red, have a red sensation. And there's probably memories like that too, memories of what it's like to experience grief or sadness or joy. And then there's knowledge by acquaintance, knowing someone, like your children know you. I don't know you, or maybe I get to know you, but I know things about you that Walter said now, but that's knowledge that. But knowledge by acquaintance is a 4th kind of knowledge, and I think there's an analog about memory. So that's just a comment. I don't know if it's worthwhile or anybody investigates those other 2 kinds of memory that correspond to those 2 kinds of knowledge.
Yeah.
The question I have 2 about the exact things you said. One is just to maybe you can give me a better example. I don't quite understand it yet about the difference between rich memory and low. I think that's the contrast you drew. And the other question is methodological about complexity memory versus not so complex. And you brought that up when there were pictures, and you said that this one picture contained complexity and the other, which was a scene of, I think, a mountain and a tree, and the other was an office with several items in it. And I don't know how you judge complexity. There's one way to do it up front and the other is from bottom up. Up front would be just say, well, let's count things in the scene. And, oh, that one has more things, this one has fewer. That's not so precise. The mountain might have a lot of complexity in it. It might have a bunch of boulders and snow on it and patches and stuff like that. So I think it's kind of hard to make this distinction. But a bottom-up way of doing it might be, we'll just give pictures to people, children of various ages, and they do really well, the young children on these kind, and they don't do so well on these kind. And then bottom up, go say, oh, well, then what's the difference? You know, we notice there's a difference. Let's see if we can sort of name it or categorize it and like that. And I don't know how you proceed, whether it's bottom up or you have a preconceived idea of what complexity is. So that's my question of methodology. The other one's about, could you give me say more about what you mean by rich memory?
Yeah, and I'm really thankful for you asking the question, because I kind of spend a little bit more time there, because I think this is the audience to have that discussion about that more. What does it mean? So I start with the first question. I start with the first. Rich is just, I kind of put it as a placeholder. I don't buy too much into it, but the 2 ways by which we can think about these developmental distinctions that we do see in the data. Some aspect develop, measures that we have, no aspect. Some behavioral measures that we have from the task that we use, right? We're always limited by that when we look at measurement. Seem to be fairly stable with age. The older you are, you do it better. And of course, with longitudinal data, we can actually maybe look at developmental change in those aspects. This is really just about how we can measure memory. And now we can look at what is the difference between those types that stable with age and those that don't. My synthesis across a wide range is that the minute you have single items versus associations between items, you find this distinction. The minute you have, this is kind of on the one end, the minute you, the kid or we're able to ask, how many details do you remember? If there's not many details, that is a type of memory that doesn't show different. You remember many details, it goes up. If we ask, do you really remember? Tapping into this in memory is called recollection. We find this difference. If we just tell them, does it just look familiar? Tapping into what in cognitive neuroscience called as a no, K-N-O-W, no differences with age. So there's a range of phenomena that are all, all very different, and we can spend a lot of time thinking about exactly how they're different. But for the sake of us just being to communicate, I like the word rich, but I acknowledge it's not a very descriptive, it's not a very good word to capture all of those different variations. So thank you for the opportunity to kind of walk you through those specific examples. And what is similar across the examples that I've shown you now is that if we can think about the representations, the content that is being represented, those are representations of what I would say high level of dimensionality and low level of dimensionality. So this is kind of where we can think about the differences in the representation, but it's not just this, it's also the processes. So the ability to bring additional strategies, for example. When you need those.
Say that again. Will you say that again? Bring additional what? I didn't hear it.
Strategies. So kind of sort those words together in order by their semantic relevance, right? So all those are different aspects, and we're still pursuing it very much. And Sijing Yu in her dissertation has specifically been doing a lot of thinking about how should we capture the different dimensions in memory in a way that we can pinpoint which of those differ by age and which of those don't. Okay, and is it about the representations? And is it about the operations? And of course, those operations act on representations, and it gets very complicated very quickly. But this is kind of how we start to try to pull out different aspects. So this is a point one, very well taken, really important. Point two, for the specific way in which in our scenes we define complexity, high and low complexity. I zoomed over because we didn't have much time to spend. But in order to do this, I went to a scene perception expert, and had the discussion. What would you describe as high or low complexity? Many different ways. You can look even just within this. I think not even within this bottom up suggestion that you have. You can look at the spatial frequency. Like, do you have a lot of differences, or there's more contiguous surfaces? More of this, there's more information in the spatial frequency of the scene itself, right? So you can do this, and you can do it on many different aspects. With discussions with her, we realized that, or her intuition was that looking at the number of unique object categories seemed to get at the best place where you get something about the complexity, but it's more likely related to what you would think of as a top-down. Because the way you see a scene, if it has 100 chairs, you don't start seeing each individual chair, you just say there are chairs. Right? So we kind of took these unique object categories and said, we're just going to give it a try. Interestingly enough, by the virtue of looking at the developmental dissociations in the memory performance, it seemed to give us some indication of a top down, or not the bottom up, like the way individuals, right? Because it ended up being that specific choice of complexity, the low complexity didn't differ by age, the high complexity did differ by age. So there's something about our choice of that specific way to, again, pull a thread of what is it that in the phenomenology of how you encode information that makes sense. So this is what I have to say about that. And of course, there's many different, like, even in this example, we started by saying, let's split the indoor versus the outdoor, right? Or we can look at those scenes and actually think about how open scene and closed scene. There's a lot of different ways in which we can probe it. For us, that high complexity, low complexity seem to really hit, we kind of hit a good spot there, allowing us to get something in how we define it. We actually went and used this to identify, to kind of draw those different, count how many different items. But it's really interesting. Just to say one other last word on this. Now with machine learning algorithms, you can actually get for each scene that is out there an algorithm to tell you how likely this scene is to be remembered. A memorability index based on machine learning algorithm that you can't quite understand too much about what it is. But and now we haven't done this kind of work yet, but we could go and say, look, this is likely to be remembered, be more or less memorized, and let's see how the brain reacts differently. Thank you.
It's interesting that it looks like we categorize according to objects rather than properties. So I was thinking a mountain might have many properties, snow patch here, dark here, light there, but those don't count as extra things according to the way we categorize. Apparently, there's some kind of gross categorization which seems to be natural rather than, I don't know, other kinds of things
And
introduce complexity.
Right. I think this is a lot of how we tend to, it's our intuitive way of thinking about it. But there are some properties, as I, for example, described this openness. Do you see the horizon, or do you have a wall? That's a property that also seems to be relevant. And that, just as you, is what I was reminded you were also asking about there's different types of knowledge. And of course, there's a lot more we can talk about. And I'm happy to hear from you a bit more. But certainly there are a lot more that's done in cognitive neuroscience. Emotional memory, what exactly is emotion, and how emotion change memory, and whether it's something about a state or a specific process that kind of tags certain memories that are more emotionally salient or not. Those are all very important aspects. And so I'm happy to talk about, this is an example of the number 3 of the types of knowledge that you mentioned, but I'm sure we can have a really interesting discussion on the others. So thank you again.
Thank you.
All right, so I think Dr. Edward has some last words and we'll do some closing remarks.
Just to thank Noah for a wonderful talk, very important talk. As a linguist, I was trying to follow it and to have some information about how my own memory works. And so it was very, very helpful. And thanks for inviting her family here. I saw a couple of people whose last names were Ofen as well. So, and I saw people whose spelled their names with a different orthography. So it seems as if they were from Israel. So Thank you all. Thank you for bringing an international audience to this talk. And so once again, I'm so grateful to the scholars in the psychology department. We've had now, I think so far 3 or 4 people in our particular series this year, and each one has given us a fantastic talk. So thanks again to the department. So Kennedy, why don't you close things off for us?
Yes, I'm not going to keep you too long, but I think this was a really wonderful talk. It was well crafted. I'm so happy we were able to have you on, and we definitely will be reaching back out to you because I think this is some important information. And the fact that we still have individuals still here afterwards, I think you facilitated some really meaningful conversation, and I'm happy we were able to do that and host you. And as a small token of the Humanities Center's gratitude, we do have a couple gifts that we give to all of our speakers. It's a journal and it's a mask, and we wish we could give this to you in person, but we'll be reaching out to you towards the end of the school year to safely get these gifts to all of our speakers. So look out for your email. Thank you again, and thank you to our wonderful audience for still staying on. This was a great talk, and we're glad to be able to do this. So if that's it, if you have any last words from our speaker, then we can close out.
Just thank you, everyone. Thank you for inviting me again, and thank you, everybody, for your attention.
All right. Well, thank you all so much, and everyone stay safe. We do have another talk tomorrow, same time, same link. You guys have a wonderful